Hello children, welcome to aimsindia's online classes. This is biology session. You are watching reproduction, the generating system, part 8. So in the last class, the basics of cell division we have discussed. Cell division is an essential process in all living organisms during which each parental cell divide and give rise to two daughter cells, which again grow, divide and give rise to new population. So what is cell cycle, children? Cell cycle is the period from the end of one division of a cell to the end of next division. It is comprised of two main stages: is interphase and M phase. M phase is a phase, we also call it as prophase, metaphase, anaphase, and phase. Okay, children. So let us discuss more about this cell division and cell cycle in this video. Let us proceed. Cells in such as heart and brain an individual never divide. So this is an interesting feature you need to remember. As soon as these organs are formed, which one? Heart and brain. So the cells of these organs once differentiated. Actually, the growth of an organism involves cell division, cell differentiation, and cell maturation. Once again, followed by cell division, cell differentiation, cell maturation. Like that it will continue. But peculiarly in human beings, some organs like heart and brain, once the cells are differentiated, they stop dividing. Okay, they never divide. So that's why if any damage occurs to these parts, will be the permanent damage. Okay, on the other hand, the bone marrow cells, bone marrow. What is bone marrow? The tissue which is present inside the bones. The bone marrow cells actively divide cells which have a short lifespan. Oh, you have studied 120 days is the lifespan of RBC. After that they die. So there is a need for again formation of new cells. That's why the bone marrow tissue acts as erythropoietic organ, the organ which is blood. For example, if you cut your finger and bleed, soon a blood clot forms to stop the bleeding. So this brings in various chemicals to the site that stimulate the skin cells to divide and heal the wound. So wound is another interesting process. Actually involves mitotic cell division. Mitotic cell division. Okay, where the edges of the wound, the cells which are located, they undergo the mitotic division and they fill the wound. It is called wound healing. Cell division increases as the wound is completely healed. In contrast, if what happens if actually this is a controlled process, highly controlled process, or regulating process, regulated process. If it loses its regulation, then what happens? Cells produce cancer. Okay, in contrast, substance, both and continual dividing cells, that ultimately host cells. For example, monocyte, one type of UBC, continue to produce, continue to produce more in number. When their number is not regulated, then what happens? They start eating away the normal cells. That is blood cancer. Okay? So important to understand the process involved in cell division. So the cell cycle, understand cell division. Okay? So cell cycle is and mitotic phase. What is cell cycle? The process of cell division is called mitosis. Active cell division. The period between two cell divisions is called interphase. What is interphase? The period between the two cell divisions is called interphase. Okay, it is the longest phase. About 96% of the cell cycle, which cell prepares itself and performs replication of genetic material. It is into three, that is G1 phase, S phase, and G2 phases. In general, the interphase is further divided into three subphases. So in G1 phase, that is growth phase 1, the period of maximum growth takes place at the end of the cell. Okay? During this, what happens? RNA and protein synthesis occur. The duration of this G1 phase varies from cell to cell. Followed by next S phase. S phase is also called synthetic phase. It is also called synthesis phase. DNA replication occurs. Okay, it may be in 7 hours. It may be. G2 phase. G2 phase is the next phase, or the phase is the interphase. Growth phase. And the macromolecule synthesis, that is carbohydrates, proteins, amino acids. They are volume of it may last long to 5 hours. After this interphase, then the mitosis phase occurs. The mitosis phase is very limited. It occurs only 60 minutes. That's all. But maximum the cell spends in interphase. So interphase is actually the period when the genetic material makes its copy at cells during mitosis. That's why it is the preparatory phase. The cell prepares for cell division during the phase. The interphase can be divided into three phases. G1 phase, S phase, and G2 phase. G1 phase the longest, is 10.5 hours. Is also then G2 only 3 to 1 hour. Okay. Next, what is G1 phase? It is this is between the completion of mitosis and the beginning of DNA replication. So that's why this is also called gap 1 phase. G1 means gap 1 phase. The cell size increases during this period. That's why 1 also we can also call this. Okay. S phase. S phase means this is the period of DNA synthesis, or it is also phase leading duplication of chromosomes. Between the end of DNA replication and the beginning of DNA replication and the beginning of mitosis. Phase 2, growth phase 2, and prepare chromosomes for mitosis. M phase, interphase followed by M phase. This is nothing but mitotic phase, mitotic division phase. Okay. G1 on cell growth, growth phase 1. It is also called the interphase. Interphase divided into 3 phases, T1, S and T2 phases. So in S phase, DNA synthesis synthesis phase. The next G2 phase, cell growth companies get prepared for the okay. Then G2 after the interphase will finish and start the mitotic phase. Mitotic phase is again divided into the karyokinesis and cytokinesis. Mitosis means nuclear division. Then cytokinesis. By the end of mitosis, two daughters are formed. Okay. So let us observe the different stages of mitosis. What happens during the mitosis? Okay. So one thing you have to remember: phase. So some cells such as nerve cells and even RBC neurocytes that do not divide further during the cell cycle, they exit from G1 phase to enter into an inactive stage. It is called as a quiescent stage, or it is also called G0 phase. G0 of the cell cycle. So now let us proceed to M phase, the phase of actual cell division. Take a permanent slide. Shows the different stages of mitotic cell division from your draw the diagrams in your and compare the division of the plasma is called as cytokinesis. During the cells undergo division, it is not easy to differentiate different stages of division. Okay, first mitosis. So the mitosis consists of karyokinesis, division of nucleus, division of cytoplasm. What is karyokinesis? The karyokinesis occurs definitely in meiosis, definitely in meiosis and mitosis, whereas cytokinesis occurs in common manner. Okay. So observe here, the mitotic phase is divided into 4 phases, or mitosis can be completed in 4 phases. That is prophase, first one, then metaphase, anaphase and telophase. So among these 4 phases, prophase is the longest one. Okay. So during interphase, the cell prepares for the division. Now nucleus starts dividing. So it is called as karyokinesis. Karyokinesis can be studied under 4 subphases: prophase, metaphase, telophase. So let us what happens during prophase. During prophase, contract, spiral and nucleoli become material chromosome. The chromosome which determines chromatids. The nucleus starts divide the form between start cells of the same. Different in animal cells, centrosome is present with, whereas in plants directly at the time of cell division. No pairing of chromosomes, mitosis, meiosis. So here only the formation of chromosomes takes place in the prophase. Okay. What are the changes occur in the prophase? The chromosomes contract, spiral, and become visible. Okay, you can see the chromosomes even under the light microscope. And the chromosomes split lengthwise, and the chromatids are formed, daughter chromatids are formed. They are connected by the centromeres. The nuclear membrane disappears. Breaks and release the centrioles. Centrioles reach the opposite poles of the nucleus and they start forming the spindle fibers. Now metaphase. The chromosomes move to the spindle equator. Okay. Centromeres attach to the spindle fiber. Centromeres separating the chromatids. So during metaphase, the chromosome line up at the equator of the spindle or equatorial plate. In animals, it is called equatorial. Okay, anaphase. In anaphase, the sister chromatids of each chromosome start moving to the outer pole. This chromosome takes place due to the division of centromere with the attached chromatids, means splitting chromosomes. Which takes place towards the poles. In anaphase, the splitting of chromosomes. Okay, the last one is telophase. The telophase, at each pole, the nuclear envelope is weak. But in telophase, the formation of nuclear membrane and becomes invisible. The replication contraction of chromosomes takes place in the prophase. Okay, but here decondensation takes place. Then the nuclear membrane pinches into form daughter cells in animals, or new cell wall material becomes laid down across the spindle equator. The nucleus divides into two and the division of cytoplasm. Okay. So what are the changes occur during telophase? Okay, in each and two daughter nuclei are formed by the end of a telophase. Okay, telophase is followed by. So after the formation of two daughter nuclei, what happened? The total cytoplasm present in the parent cell equally distributed to the two daughter cells. Mitosis. Okay, read the textbook and take notes. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.